In this video, we'll be looking at the cardiovascular system, and in particular the issues around blood flow. Which components make up blood flow? Now, why are we so concerned about blood flow and blood pressure? Well, think about it. Anytime you go into the hospital or emergency room or to visit a doctor, many times they'll be taking blood pressure, and because looking at blood pressure gives us an idea of the amount of perfusion tissues receiving. Now, what is perfusion? Perfusion is the amount of blood that a given amount of tissue receives in a given amount of time, usually within a minute. Now, many tissues in the body are highly sensitive to changes in perfusion. The brain, the heart, the kidney, very metabolically active tissues really require constant blood flow. Now, don't be alarmed by looking at this flow chart to start with. We're going to break it down as we go, but this is one of the most important pieces of paper you'll receive in this course and have a look at. This gives the major factors that have an effect on blood flow. So we're going to start off by talking a bit about arterial pressure and just the concept of flow. Now, before we go too far, I think we should take a few minutes and really think about the cardiovascular system. The cardiovascular system is a closed system in that it's a continuous loop. And this continuous loop is made up of a number of components, and each one of these components really is a separate chamber. Now, what I've done here is over on this side, you see that there's two squares. These are two chambers that are interrelated. This is really the right side of the heart. We have the right atrium and the right ventricle. Of course, blood goes from the right ventricle to the lungs, which is a chamber into itself if you look at blood flow because it has a number of blood vessels. Blood goes from the lungs once it's oxygenated, hopefully, to the left side of the heart, the left atrium, the left ventricle, and from the left ventricle, blood gets pumped out into the arterial system. And we can see the arteries here. Now, of course, I've overly simplified it, that as we move further down, we'd go from one artery to two to four, and the numbers increase quite a bit. Arterial blood then goes into the capillary system. And once again, I've shown just a few here. From the capillary system, blood now enters the venous system. By the time the blood gets through the capillary system, blood pressure has dropped quite a bit, and we'll come back and talk a bit about the concept of blood pressure and the factors. Now, let's get back to this thought about it's a series of chambers, this closed system. We can clearly see there's chambers up here. This is another chamber, chambers. This can act as one big chamber. But when we get back to the the venous system, we can see that we actually start to have valves. Here's a close-up of inside a vein. You have valves, and the valves create a series of chambers where blood can move from one chamber to the next. So, just a reminder, is that the cardiovascular system is a closed system. It's a series of chambers. And really, how does blood move from one chamber to the next? It's the result of pressure differences. So let's have a quick look at that. Here's two chambers. This could be, let's say, the left atrium and the left ventricle. Well, how does blood move from one to the next? Well, it really is based on pressure, as I said. Fluids and gases move from an area of high pressure to low pressure, as we can see here. We increase the pressure in the first chamber. This pressure is greater than the pressure in the second chamber, and material will move. We see this happening throughout the body. The cardiovascular system, blood moves from one chamber to the next. This is also the same thing that happens in your lungs, but instead of with a fluid, it happens with a gas. Now, let's ask, how can we increase the pressure in one chamber to make it move? Well, one of the ways we can do it is we can keep the chamber the same size, but simply add more material to it. If we can force more material into this chamber, the pressure will go up. And once the pressure's gone up high enough, stuff will flow from here into here. That's the first way. There's a second way we can do it, and that is we can reduce the chamber. If I keep the same amount of material that was originally in the chamber, but now make the chamber smaller, the volume has gone down, all of a sudden pressure goes up. So as I decrease volume, and I don't allow anything to escape at that point, pressure will go up. Now, consider, where do we see this happening in the body? Probably doesn't take you too long to figure out this is what the ventricles of the heart do. This is also associated with respiration. As I breathe out, the lungs recoil, pressure builds up, and forces air out. Alright, now that we've talked a bit about stuff moving, let's come back to this, what looks like a scary but very, very useful flowchart. Everything here that we see up here leads to or affects arterial pressure or flow. The first two components is cardiac output, and we'll come back look at that in a minute, and peripheral resistance. Peripheral resistance are the factors that oppose the flow of blood from one chamber to the next or through something like a blood vessel. So let's start there. Peripheral resistance has three main factors. The first one is vessel length. The longer a blood vessel, the more resistance there will be. Okay? So we don't change the diameter, don't do anything, but if blood has to flow through a short blood vessel or a long blood vessel, the longer blood vessel basically results in more resistance. It opposes the movement of flow. The second resistance factor is vessel diameter. This is the width of the blood vessel. And what it comes down to is that the wider a blood vessel is, the less resistance. So if I'm trying to send blood down two different blood vessels, one is very narrow and one is very wide. The wider vessel, because it has a greater diameter, offers less resistance to the flow. The third thing that we can look at that affects total peripheral resistance is viscosity. How thick? Think about fluids. Water versus honey, right? That gives you an idea of viscosity. The thicker the fluid, basically, the more resistance. So, let's ask ourselves this question. Which of these three things, vessel length, vessel diameter, and viscosity, can we change? Vessel length, no, can't change it. It is what it is. It's not like I can shrink my arm, make it 6 inches shorter if I want to reduce the resistance of blood moving to my fingertips. Now, vessel diameter. This is a really important one, because what we'll see in further lectures is that a small change in vessel diameter has a very large effect on resistance. And of course, resistance has a role in how the blood will flow. The next thing is viscosity. Can I change viscosity? Yeah, we can. Some of the ways we can change blood viscosity is, let's say, dehydration. You don't have as much water in your body as you should. The blood is largely water. Therefore, you have less water in the blood, and you still have the same number of red blood cells. The blood looks thicker and is thicker. The other way we can increase viscosity is by increasing the number of red blood cells. Let's say you move from Vancouver to Denver, Colorado at high altitude. Your kidneys will sense this. It'll release a hormone called erythropoietin, and you'll make more red blood cells, and your blood becomes thicker. So, vessel length we cannot change. Vessel diameter we can change very quickly and has a very profound effect. Viscosity, yes, it can be changed. It tends to be a slower change. Now let's look at cardiac output. And cardiac output is made of two factors. Number one is heart rate. Most of you know this. This is beats per minute. And it makes sense. If my heart is to beat faster, then more blood will come out of the heart every minute. The next thing that affects cardiac output is stroke volume. Now let's define stroke volume. Stroke volume is the amount of blood that leaves, let's say in this case, the left ventricle. So that's usually measured in milliliters. So cardiac output, so here's cardiac output down here, is a function of heart rate times stroke volume. And you'll often see that written out like a mathematical equation. Cardiac output equals heart rate times stroke volume. Okay, now we can ask ourselves the question, well, how can I play around with stroke volume? How can we change it? Well, there's two major things that affect the amount of blood that comes out of the heart and can affect the number of milliliters. One group of things we can refer to as diastolic function. The other is systolic function. Now, diastolic refers to basically relaxation. And think about it. If we have a chamber, say the left ventricle, and I want to have blood come out of it, it's not only the contraction of that chamber, it's also the ability of it to fully relax so it can eventually fill. And diastolic refers to the relaxation. There's two major things that can affect diastolic function. So here we are, we're on diastolic function. The first thing is the pericardium. Now, if you remember back to our basic heart dissection, or the dissection we'll do in the future, you'll have a look at this, is the pericardium is a sac on the outside of the heart. It's fairly fibrous. Now, this pericardium, if it's scarred or if there's fluid buildup between the heart and the pericardium, this is really going to limit the amount that those ventricles can expand or relax, which will eventually have an effect on how they fill and eventually on stroke volume. And eventually, we're following our way down the flow chart to cardiac output and blood flow. So issues with the pericardium can affect diastolic. We also can have issues with compliance. Now, what's compliance? This is the way I think of it. It's the stretchability, and I'm going to probably make up a word here, reboundability of the heart tissue. If, say you had a heart attack, a myocardial infarction, you may now have scar tissue within that heart tissue. It doesn't expand and it doesn't contract the way it's supposed to. So we need that beautiful expansion and relaxation. So if there's issues with that, you may not get a full relaxation and can affect diastolic volume. Let's move over to the last group of things, and that is systolic function. So if diastolic function was the relaxation, systolic is the contraction. And we're going to look at the last three items associated with it. Number one is preload. So there's preload. Preload is the amount of blood in the ventricle just before it contracts. Okay? So if I'm able to put more blood into a ventricle before it contracts, that means I'm probably going to have more blood coming out of that ventricle, which would then increase the stroke volume, which would then increase the cardiac output, which would then increase the flow. The next one is contractility. And it's what you think it is. It's the ability of that heart tissue to contract. If there's something like, as I said earlier, maybe there's scar tissue from a heart attack or myocardial infarction, may prevent that contraction from happening. That would have an effect. Or there are certain things in the body that help to increase the ability of the heart to contract. And once again, we'll see that in other lectures and other talks. So contractility is a big one. Let's go to the next one, the third of this group, and that's the afterload. Okay, here comes the big warning. Now, we talked about preload, and we said preload is the amount of blood in that ventricle before it contracts. It's very easy to fall into this trap and think that afterload must therefore be the amount of blood left in that ventricle after contraction. Big warning, flashing signs. It isn't. Okay? Afterload actually refers to the pressure in the next chamber. Okay, let's take a second. Remember we said the cardiovascular system is a series of chambers that are interrelated. Let's take two of those chambers, the left ventricle of the heart and let's say the aorta, where the blood moves from the left ventricle into the aorta. The afterload we'd be looking at would basically be the pressure in the aorta. Now remember, stuff moves from one chamber to the next based on pressure. So let's say my aorta, the blood in my aorta or my arterial system had a high pressure. That means the left ventricle of the heart, which is trying to push the blood into the aorta, is going to have to create even more pressure in order to move the blood. So a high or an increased afterload has an effect on your ability to move blood from the first chamber to the second. For an example, a high afterload is something which is very common, and that is hypertension. That if you have hypertension, it means that the left ventricle has to work even harder, and that can lead to a number of issues down the road. All right, so this is a very quick overlook, and we'll be spending time in quite a bit of time in following lectures and talks going through the interrelationships between these different things, things like afterload, stroke volume, cardiac output. But this is a fabulous one-page diagram that you should commit to memory as soon as possible, because this is really the backbone of understanding blood flow and blood pressure. And blood flow and blood pressure is probably one of the most important concepts when we're looking at human physiology. All right, thanks for your time, and we'll be chatting soon.